Saturday, September 9th, 2023, AGNDD Intellireport topic, Neurotransmitter. Please pardon any visual and informational inaccuracies in this entirely AI-generated Wikipedia-sourced video article.
This text provides an overview of neurotransmitters, including their synthesis, classification, and storage. It also highlights the different types of neurotransmitters and their origins. Neurotransmitters are synthesized in neurons and are derived from precursor molecules found in the cell. There are different classes of neurotransmitters, including amino acids, monoamines, peptides, purines, and metabolic products. Monoamines are synthesized by modifying a single amino acid, such as serotonin from tryptophan. Peptide transmitters, or neuropeptides, are protein transmitters that often work together with other transmitters. Purine neurotransmitters like ATP come from nucleic acids, while others like nitric oxide and carbon monoxide are metabolic byproducts. Neurotransmitters are usually stored in synaptic vesicles near the cell membrane at the axon terminal. However, certain neurotransmitters like carbon monoxide and nitric oxide are synthesized and released immediately after an action potential without being stored in vesicles. Neurotransmitters are chemicals that are released in response to electrical signals in the presynaptic neuron. They can also be released at a low level without stimulation. These neurotransmitters then cross the synaptic cleft and bind to receptors on the postsynaptic neuron's membrane. Neurotransmitters are released into the synaptic cleft and diffuse across the synapse to interact with receptors on the target cell. The effect of the neurotransmitter depends on the receptors present on the target cell. Binding of neurotransmitters can lead to excitation, inhibition, or modulation of the postsynaptic neuron, depending on the type of receptor. Neurotransmitters are removed from the synaptic cleft through diffusion, enzyme degradation, and reuptake. Diffusion involves neurotransmitters drifting out of the cleft and being absorbed by glial cells. Enzyme degradation occurs when proteins break down the neurotransmitters. Reuptake involves neurotransmitters being reabsorbed into the presynaptic neuron through transporters. This process helps maintain proper synaptic function. Cocaine, for example, blocks the reuptake of dopamine, leading to prolonged activation of dopamine receptors. Scientists used to believe that most communication between neurons in the brain was electrical. However, in the early 20th century, Ramon Y. Cajal discovered a small gap between neurons called the synaptic cleft. This suggested that communication between neurons occurs through chemical messengers. Otto Loewy confirmed this in 1921 by showing that neurons can communicate by releasing chemicals. He also discovered acetylcholine, the first known neurotransmitter, and demonstrated its role in regulating cardiac function through experiments on frog vagus nerves. To identify neurotransmitters, several criteria are considered, including synthesis, release and response, experimental response, and removal mechanism. Advances in pharmacology, genetics, and chemical neuroanatomy have expanded the definition of neurotransmitters to include chemicals that carry messages between neurons, have a common carrying function, or send reverse direction messages. Immunocytochemical techniques are commonly used to determine the anatomical localization of neurotransmitters by identifying the location of transmitter substances or enzymes involved in their synthesis. Various techniques and experiments can be used to identify neurotransmitters in the central nervous system. Neurons communicate through synapses, where neurotransmitters transmit signals. When an action potential reaches the presynaptic terminal, neurotransmitters are released into the synaptic cleft. They bind to receptors on the postsynaptic membrane, influencing the receiving neuron. If the excitatory influences outweigh the inhibitory ones, the receiving neuron may generate its own action potential, continuing the transmission of information. This process allows for information flow and the formation of complex neural networks. Neurotransmitters can have excitatory, inhibitory, or modulatory effects on target cells based on the receptors they interact with. Excitatory neurotransmitters increase the likelihood of an action potential, while inhibitory neurotransmitters decrease it. Type 1 synapses have excitatory effects and are located on dendrites, while type 2 synapses have inhibitory effects and are located on the cell body. Modulatory effects are spread throughout all synaptic membranes and can result in changes in sensitivity to future stimuli. The different locations of synapses divide a neuron into an excitatory dendritic tree and an inhibitory cell body. Neurotransmitters are chemical messengers in the brain that activate receptors and have various effects on the body. Glutamate is an excitatory neurotransmitter involved in memory storage, but excessive release can lead to cell death. GABA is an inhibitory neurotransmitter that helps regulate brain activity. Acetylcholine activates muscles and operates in different regions of the brain. Dopamine regulates motor behavior and pleasure. Serotonin regulates various functions like appetite and mood. Norepinephrine stimulates the release of adrenaline, and epinephrine plays a role in the fight or flight response. Neurotransmitters can be classified into different categories, including amino acids, peptides, monoamines, gasotransmitters, purines, and others. Some major neurotransmitters include glutamate, GABA, dopamine, serotonin, histamine, and acetylcholine. Neuroactive peptides are also important neurotransmitters, and over 100 have been discovered. Some ions and gaseous molecules, such as zinc, nitric oxide, carbon monoxide, and hydrogen sulfide, are also considered neurotransmitters. Glutamate is the most prevalent excitatory neurotransmitter, while GABA is the most prevalent inhibitory neurotransmitter. Many psychoactive drugs act by altering the actions of neurotransmitter systems. Certain neurons that produce specific neurotransmitters can form distinct systems in the brain, known as neurotransmitter systems. These systems, such as the noradrenaline, dopamine, serotonin, and cholinergic systems, have a widespread impact on the brain when activated, a process called volume transmission. Trace amines, through their interaction with trace amine-associated receptor 1, modulate neurotransmission in monoamine pathways, dopamine, norepinephrine, and serotonin, throughout the brain. The following is a brief comparison of these neurotransmitter systems. Research on the effects of drugs on neurotransmitters is a significant focus in neuroscience. Drugs can alter neurotransmitter activity by affecting synthesis, release, storage, binding, deactivation, or action potential. Some drugs decrease neurotransmitter synthesis, while others block or stimulate release. Drugs can also act as receptor antagonists or agonists, mimicking or preventing neurotransmitter binding. Additionally, drugs can interfere with deactivation or block action potentials. Understanding these effects can help in treating neurological diseases and disorders. Examples of drugs include cocaine, fluoxetine, AMPT, reserpine, and deprenyl. An agonist is a chemical that can bind to a receptor and produce the same response as the natural substance that binds to that receptor. Agonists can act directly by binding to the receptor site or indirectly by increasing the release or preventing the reuptake of neurotransmitters. Examples of agonists include nicotine, which binds to nicotinic acetylcholine receptors, and opiates, which bind to mu-opioid receptors. Amphetamine is an example of an indirect agonist that stimulates neurotransmitter release and prevents reuptake. An antagonist is a chemical that reduces the activity of another chemical substance in the body. There are two types of antagonists: direct acting, which block receptors and prevent neurotransmitters from binding, and indirect acting, which inhibit the release or production of neurotransmitters. Examples include atropine as a direct-acting antagonist and reserpine as an indirect-acting antagonist. An antagonist drug is a type of drug that binds to a receptor without activating it, preventing other substances from binding to and activating the receptor. There are two types of antagonists: competitive and irreversible. Competitive antagonists compete with agonists for binding to the receptor, while irreversible antagonists bind strongly to the receptor, rendering it unavailable for agonist binding. Both types of antagonists decrease the physiological response and require higher concentrations of agonists to produce the same response. The effects of increasing neurotransmitter precursors on neurotransmitter synthesis, release, and receptor firing are not fully understood. While it may increase neurotransmitter synthesis, it is uncertain if this leads to a long-term increase in signal strength. The nervous system can adapt to changes and maintain constant firing. Some neurotransmitters may be involved in depression, and there is limited evidence suggesting that taking precursors of these neurotransmitters may be beneficial for mild to moderate depression. L-DOPA is used to treat Parkinson's disease, as it can cross the blood-brain barrier and become dopamine. However, there is limited evidence supporting the use of neurotransmitter precursors for depressed patients with low norepinephrine activity. L-phenylalanine and L-tyrosine can be converted into dopamine, norepinephrine, and epinephrine with the help of vitamin B6, vitamin C, and S-adenosylmethionine. Some studies suggest potential antidepressant effects of these precursors, but more research is needed in this field. L-tryptophan, a precursor for serotonin when administered, can double serotonin production in the brain and is more effective than a placebo in treating mild and moderate depression. Vitamin C is necessary for this conversion. Additionally, 5-hydroxytryptophan, 5-HTP, another precursor for serotonin, is also more effective than a placebo. Diseases and disorders can impact neurotransmitter systems, resulting in different imbalances. For instance, a shortage of dopamine can lead to Parkinson's disease, while an excess or deficiency of dopamine might contribute to schizophrenia or ADHD. Depression has been associated with serotonin levels, which has led to the use of selective serotonin reuptake inhibitors, SSRIs, to boost serotonin levels. Glutamate imbalances have been tentatively linked to conditions such as autism, OCD, schizophrenia, depression, and neurological diseases like Parkinson's, multiple sclerosis, Alzheimer's, stroke, and ALS. There are no established norms for neurotransmitter levels, and it is difficult to measure them accurately. Imbalances in neurotransmitter regulation can affect temperament in healthy individuals and are associated with various diseases and mental disorders. Chronic stress and genetics can contribute to changes in neurotransmitter systems. Medications that interact with neurotransmitters are commonly prescribed for psychiatric and psychological issues, but the evidence supporting their effectiveness has been criticized. Dopamine imbalance has been linked to neurological disorders like multiple sclerosis. In conclusion, neurotransmitters play a crucial role in communication between neurons. They are synthesized from precursor molecules and can be classified into different categories. While most neurotransmitters are stored in synaptic vesicles, some are synthesized and released immediately.
Thank you for watching this AGNDD Intellireport. Like, subscribe, and click the bell button to get notified during new uploads. And make sure to tune in next time. Stay safe and healthy. Cheers.